And today we're kicking off with our very own Anna Rentsch, who is a postdoc here at University of Guelph in the Betacarts lab, and she's going to tell us about feed restriction, frustration, and locomotion in chickens.
Good morning, everyone. Thanks for making it here so early. Okay. I will be talking about laying hen pullets. For those who don't know, the pullet is the immature or adolescent chicken, and the laying hen is the chicken that has been selected for egg production. And therefore, it's not surprising that research and genetic selection has mostly been focusing on egg parameters. So we want more eggs, we want them to be laid earlier, we want them to be laid for longer. In the laying hen industry or in the egg industry, puberty is traditionally triggered with photostimulation. And what that is a lengthening of the day or the light period in the barn, kind of simulating spring. And that triggers the onset of puberty in the chickens. However, we have also in recent years noticed that there also appear to be metabolic thresholds. So once a pullet reaches a certain body weight, she will also go through puberty, even if she has not been photostimulated. And another thing I would like to stress here is that egg production is also influenced by stress. So if you have extreme or chronic stress, the stress axis can actually negatively influence the reproductive axis or the HPG axis. And I'm specifically going to be talking about feed restriction in laying hen pullets. Now, the poultry people amongst you are probably going to be like, what? Why? Because feed restriction is usually done in broiler breeders. These are the parents of our meat birds, and they are quite severely feed restricted during development, so that even despite their selection for accelerated growth, they can still reach a mature age. Now, in broiler breeders, we feed restrict them during development anywhere between 50 and 90%. So they get to eat what's left over of that. And we know that this is a welfare issue. We know that these pullets will show redirected feeding behavior or abnormal feeding behavior. They might peck things that are not food. We know that they show increased aggressions. So we know this is an issue and this has been well studied. However, in laying hen pullets, feed restriction during development can also be used as a management tool to have a more uniform body weight in your pullet flock. So you can then avoid having eggs in the pullet barn. So you can avoid having these pullets that are a bit heavier than everyone else for some reason, already reach that metabolic threshold and already start laying, even though they haven't been photostimulated. So feed restriction can help with that. And in research, particularly, feed restriction can obviously be used as a treatment to slow down growth trajectory, and it can be used to delay onset of lay. Now, the feed restriction that we do occasionally use in laying hen pullets is not quite as severe. Scanning the literature, I found amounts of 10% restriction up to the highest I've seen was 50%, but usually somewhere around 20. So it's not quite as severe as in broiler breeders, and that might be one of the reasons why, even though laying hen pullets are occasionally feed restricted commercially and a bit more often in research to modify growth trajectory and age at first egg, there really isn't any research on whether this is stressful for these birds. And that's important for two reasons. One is, if it is stressful and our outcome variable in our research are egg production traits, well, that would be a confound, right? So it's bad for research if that was really stressful treatment. And on the other hand, and the thing that would affect or bother me more is if it would actually affect the welfare of these pullets. So going into this project, I had two hypotheses. One is feed restriction during development might lead to frustration. And if it did, we will probably see altered feeding behavior as well as an increase in aggression if they're housed socially. The other thing I hypothesized was that feed restriction would alter the energy expenditure. So if the pullets are moving vertically, they might choose to use modes of locomotion that are less energy demanding. And that could, again, have impact on things like skeletal development. And I could actually make use of an experiment that was already ongoing when I joined my current lab, which is fabulous because I didn't have to subject more hens to my treatment. And the experiment that was ongoing was of a split plot design. So we had the main plot factor is age at photostimulation, here indicated with the little stars. So there was an earlier and a later time point at photostimulation. And for both of those groups, the subplot factor was having two different feeding regimens. So between the ages of 14 weeks of age and 21 weeks of age, half of the pullets were feed restricted by 20%, and they reached a 10% difference in body weight by 21. Actually, I think that was 20 weeks. And that gave us 4 different distinct groups. So there were birds that were feed restricted and they were photostimulated before they reached a metabolic threshold. Birds that were fed ad libitum and they were their photostimulation was synchronized with reaching the metabolic threshold. Restricted birds where the photostimulation was synchronized, and ad libitum fed birds that were photostimulated after. And between weeks 15 and 22, we video recorded our home pens on the weekends, which for 2 consecutive days every week. We chose the weekends because there wouldn't be any disruptions in those pens, except for the feeding of the restricted birds, which happened twice a day. So once in the morning, once in the afternoon, they would be given their allotted feed. This is what our pens look like. We had 24 of those pens. We have elevated perches in there. 2 of them, we had 2 elevated platforms. There'd be a ramp going up to the higher platform. There's nest boxes in the back, and the feeding trough in the restricted pens was long enough that all the pullets could eat at the same time. So for both of those days, we recorded 4 5-minute videos. So that's 20 minutes of total video a day, 40 minutes an age group. And we chose these video time points based on when they were fed. So an hour before and an hour after meals, the birds would be recorded for 5 minutes, and we just choose the same time for the ad libitum birds. The data was actually coded by our co-op student Sachin, who developed an amazing ethogram, and these are the different behavioral categories that we were interested in. So one is abnormal pecking behavior, so pecking at objects, pecking at conspecifics, so severe feather pecking, tissue pecking, these kinds of things. Aggression, aerial locomotion, so any kind of vertical locomotion when the birds would use their wings, we'd assume is high energy demanding. Vertical transitions using the ramp being less energy demanding. The unit for all of those behavioral groups would be the mean bout frequency per 5 minutes calculated per bird in the pen. We had to delete, or we had to exclude the data from 20 weeks of age because of technical issues. We lost a whole treatment group's worth of videos, so we decided to scrap the whole week. And Sachin actually developed and refined and retested and refined the ethogram until he reached an inter-observer reliability of a couple larger than 0.9 for all our variables before he started coding. Now, don't look at the graph yet. I first want to walk you through what all of my results slide will look like. So I always will give you all of my test statistics, so our chi-square and p-values. I will boldface everything that is statistically significant. The thing that is shown in the graph will be highlighted in yellow. You'll see my model specifics on the bottom, always below the test statistics. All of my graphs, or most of them, will have week of age on the x-axis, and then the frequency of the behavior, bouts per 5 minutes per bird on the y-axis. Okay, now you can look. So in terms of abnormal pecking behavior, we found a statistical interaction between the feeding regimens and the age. And we have our restricted fed birds in red and the ad libitum fed birds in blue. And what you can see here is kind of what we expected. So we had higher frequencies of these abnormal pecking behaviors in our restricted birds. What you also see, though, is that these differences were mainly in weeks 16, 17, and 18. And if you remember, the feed restriction was lifted at 21 weeks of age. So it seemed to normalize, or even before we lifted restriction, and definitely after that, the effect was gone. In terms of aggressive behavior, we had that same interaction. Again, ad libs in blue, restricted in red. However, this was contrary to what we expected. We actually saw a bit more aggression in the birds that were fed ad libitum. And while we were a bit confused at first, it actually makes a lot of sense if we look at our other interaction here, which is the age at photostimulation by age interaction. So while everyone's aggression increased with age, the birds that were photostimulated at 18 weeks of age had a steeper increase in that aggression than our birds that were photostimulated at 20 weeks of age. So that makes me think this might have been a hormonally driven change in aggression, and our ad libitum birds that mature earlier would have had that same increase. And we are actually planning to watch more videos up until 25 weeks of age to see if we see a corresponding peak in the group that was photostimulated later. Now, if we zoom in on only the restricted pullets, we see that both abnormal pecking behavior and aggression on the right side over there were both higher post-meal than pre-meal. So that made us think that these differences in behavior, or these effects of the feeding regimen, might not be due to hunger, but rather due to frustration, competition at having been given a meal that was not enough. So that was both for the abnormal pecking behavior as well as the aggression. And again, we have our age effects with abnormal behavior higher at 16 to 18 weeks and the aggression increasing with age. For our aerial locomotion, we had this interaction of age at photostimulation and age. Now, I personally can see a pattern here, plus none of our main effects are statistically significant. That makes me think it's probably a type 2 error situation going on here. And I don't really want to read anything into this. I'm not going to draw any conclusions based on this. Plus, our main question here was, is it affected by feeding? And it was not. And the same goes for ramp use. So ramp use was not affected by feeding at all. We again have this interaction with no significant main effects. I'm thinking this is some sort of noise that is being picked up. Now, to sum this up, so our restricted pullets showed an increase in the abnormal pecking behavior, and it was mainly higher after meals. And the effect actually disappeared even before feed restriction was lifted, but it definitely disappeared once it was lifted. Our aggression increased in all of our groups with age. It increased more so in the birds that were photostimulated earlier, and it was higher after meals than before meals. Neither our aerial locomotion nor our ramp use was dependent on our feeding treatment. So that makes me think that while feed restriction in Langhand pullets seems to induce some frustration, that frustration appears to be limited to post-feeding, and it appears to be reversible. And the feeding restriction did not alter our modes of vertical locomotion, so I don't think our birds were faint from hunger. And with this, I would like to thank you for your attention. I would like to thank the Beta Carrot Lab and all the students, definitely Sachin, who coded all those videos, the Arkel staff, the pullets, and then all of our funding agencies in Canada and the US. And I'm happy to take any questions.